Right, dear student, today we are going to talk about one of the relatively difficult topic in immunology, and we are going to make it easy. Right? The topic is major histocompatibility complex and transplantation. Major histocompatibility molecules, major histocompatibility molecules and concepts related with transplantation. Now in this series of lectures, first of all, I will discuss in detail about the major histocompatibility molecules. Once we are done with it, we have a really good and strong concept about these molecules, then we will go into detail about the transplant and transplant rejection and mechanisms involved into that. Right? So right now we will focus only on to build the basic concept, what are major histocompatibility molecules, right? Actually, in old time means just few years back, they were also called HLA molecules. HLA molecules, right? Previously they were called HLA molecules, and now they are called major histocompatibility molecule, MHC molecules. MHC molecules. So first of all, I have to explain why previously they were called HLA molecules and why now they are called MHC molecules. Is that right? And how these molecules work? What are these molecules? Actually, originally these molecules were discovered on the leukocyte, right? Initially. Later on, we found that these molecules are present. Some of these molecules are present on most of the cells on the tissues, right? Because initially these molecules were discovered on the white blood cells, the leukocytes, right? They were present on the surface of that. So initially doctor never knew what are the functions of these molecules. They just knew there are some protein molecules present on the white blood cells, especially antigen presenting cells. So doctors call these molecules as human leukocyte antigens. That is why originally these molecules were called human leukocyte antigen, which can be reduced to HLA system or HLA molecules, right? Later on, doctors discovered that some of these molecules are present on every nucleated cell. They're present on every cell on the body, some of these molecules, and they are not specific only for leukocyte. And further advances in immunology told us that these molecules determine the major tissue compatibility when you are doing tissue grafting. For example, from one person you are taking a tissue and transplanting that tissue to my body. Now presence of these molecules on the donor tissue and on the recipient tissue, right, that will determine that if these molecules are similar in the donor tissue and in the recipient tissue, then transplant will be more chances to have success. But opposite to that, if these molecules are very different in the grafted tissue and recipient, then chances of transplant success are less. So from there the concept came that these molecules do play a big role in matching the tissue between the donor and recipient, between the graft and the host. So from there this name originated, that these are the molecules which mainly determine the tissue compatibility. So these are the molecules which are the major molecules, right, which determine the histamine tissue compatibility, mean that are the tissue between the host and the graft are compatible or not, right? So these are the origin of these two words, HLA molecules and MHC molecules. Let me repeat it again. HLA molecules is the old name, MHC molecule is the modern name of the same molecules. Why they were previously called HLA molecules? Because they were originally discovered only on leukocyte. Later on, as we found that such molecules are present on many cells, especially MHC class 1 molecules are present on all the cells, nucleated cells, and we also discovered that presence of these molecule determines the tissue compatibility between the grafted tissue and the recipient, right? So we said that these molecules should be renamed as major histocompatibility molecules. So now onwards, when you study in the book that HLA gene complex or HLA locus or MHC locus or MHC molecule, they are one and the same thing. When we are talking about HLA genes, we are talking actually about the MHC genes. When we are talking about HLA molecules, actually we are talking about MHC molecules, right? So this was origin of their name, which sometimes confuses the student, right? Now we will come to what are the functions of these molecules, from where these molecules come, what they are doing in our body, what are the good things they are doing, and what are the problems they are causing to us, right? Let's go into that. What is the origin of these molecules? I will explain. Basically, there are two types of MHC molecule. There are class 1 molecules, and there are class 2 MHC molecules. Is that right? MHC molecule, class 1 molecules and class 2 molecules. Let's go into detail and study them. First of all, let's talk about, I'll discuss into detail class 1 molecule, right? Then I will talk into detail about class 2 molecule. Let's talk about, this is our typical cell. Again, let me remind you that class 1 molecules are present in all nucleated cells in human body as well as on platelets. Where they are expressed? These are expressed, these molecules, in all nucleated cells. Right? We are trying to eliminate the RBCs. Red blood cells don't have such molecules because red blood cells are not nucleated cells, right? So all nucleated cells express class 1 molecules as well as platelets. Platelets also express these molecules, but red blood cells don't have these molecules. Now we have to see from where these molecules are coming on the surface of the cells and what they are doing there, what is their importance. When we talk about these HLA molecules or MHC molecules, the first thing which should come to you is chromosome number 6. Which chromosome? Chromosome number 6. Actually, let's suppose I draw a cell here. On the chromosome number 6, how many chromosome number 6 are present in one person? 2. One chromosome number 6 is coming from mother, and other chromosome number 6 is coming from father, right? Now let's suppose this is a chromosome number 6 which is coming from, yes, from mother, right? And we have one more chromosome number 6, and this chromosome number 6 is coming from father. Is that right? So we can say this is maternal chromosome number 6 and this is, yes, paternal chromosome number 6. Now, like other chromosomes, chromosome number 6 has short arm and long arm. The genes which are related with MHC molecules, they are present on short arms, right? They are present on the short arm. Now on the short arm of this chromosome, there's a special area on which these genes are present, right? There are 3 boxes of genes here. There's one box, there's another box, and another box. This box is going to make MHC class 1 molecules. This box is going to make MHC, yes, class 2 molecule. And in between, there's another box of genes which is going to make MHC class 3 molecules. For our discussion related with immunology and tissue transplantation, today we will be concentrating on the concepts related with class 1 molecules and class 2 molecules, right? So let's see, I will draw this area in a big way, in an enlarged fashion, outside. Now, this part of the chromosome, short arm, I'm making it large so that you can really see. Now, what really happens that here is MHC locus. This was the first box, this is the middle box, and this was another box. The first box is related with formation of which molecules? Yes, MHC class 1 molecules. And this box is related with the formation of MHC class 2 molecules. Is that right? Now let's study it in detail. What is present if you open this genetic locus? What is present in it? Actually, it has 3 points. In this big box, there are 3 bags: A, C, and B. What is there? There is A, C, and B. Actually, basically, this is one gene, this is an other gene, and this is an other gene. It means that at chromosome number 6, on its short arm, the group of genes which are related with the formation of class 1 molecules, they are actually having primarily how many genes? 3 genes: A gene, C gene, and B gene. Is that right? Now, I will go into detail of just A gene. Open it and study it. How this gene is working. This is going to, let's suppose this is the A gene. You know what is gene? How do you define gene? Gene is a nucleotide sequence. Gene is a specialized nucleotide sequence which is concerned with the formation of specific RNA. Is that right? So within this whole DNA, this is a specific sequence of nucleotides which is going to make a specific RNA. Let's suppose a specific type of messenger RNA, which will be eventually translated into specific type of a protein molecule, and that protein molecule will become MHC class 1 molecule belonging to which category? A category. Is that right? Now, we will study how this gene is going to make class 1 molecule. Is that right? Actually, what happens that the nucleotide sequences in this gene in one area, right? Genes start from here, from this point up to this point. But there is a nucleotide sequence within the gene, and this nucleotide sequence keep on undergoing spontaneous rearrangements. It's just like that. Have you played cards? You know cards? Right. I sometimes think as if some enzyme is sitting there and playing cards with the nucleotides all the time, shuffling the cards. I don't know, but it is there. It's happening. Right now it is happening in every cell in your body except RBCs. Right now, what is going on? That in your every cell, in the nucleus, on the chromosome number 6, at the MHC locus, especially at MHC class 1 point, right? Or in all these boxes, A, B, and C, right? Where there are MHC class 1 genes present. In these genes, in every gene, one sequence of nucleotide is constantly undergoing rearrangements. And you know what it is doing? It undergoes rearrangement and then make a messenger RNA. Then again it undergoes rearrangement, make another messenger RNA. So what is happening? That if this gene A, MHC class 1 related gene A, if you see what type of messenger RNA is making, you will come to know this is one messenger RNA, right? After some time it will release another messenger RNA. After some time it will release another messenger RNA. Now you see a very special point that these messenger RNAs which are made by the same gene, they are having this area which is varying from molecule to molecule. When these 3 RNAs will be translated into peptide chains, let's see how are those peptide chains. When these messenger RNAs are translated into peptide chain, okay, You know, when peptide chains are made, they are folded out in a unique fashion. Now, this is the protein which is made by this messenger RNA, and it will have, suppose, a pocket here with this thing. Now the second molecule, in this case, on the third molecule, okay, let me put it slightly more different so that you can recognize. Let's suppose this was having horizontal bars in this area. This is a specific nucleotide sequence. When it will translate into protein, within the protein there will be one area which will be very much specifically related with that. Right? Now I come to this thing. In this case, nucleotide sequence in this specific part of the messenger RNA is different from the other. So when this will make a protein, this area will be same or different from other? This will be different from other. Then we come to, let's suppose, next molecule it has made, and in this molecule nucleotide sequence is different than first and second. So when it will make this one, here also nucleotide sequence will be. Now listen carefully. All of these three molecules are class 1 molecule. All of these three class 1 molecule peptide chain which is made here, they are belonging to which category? A, class 1A molecules. But there is a very important difference which you should appreciate because of the constant rearrangement in a specific part of the genes. The messenger RNAs which are made one after the other, they are having slightly different arrangement of the nucleotide in this specific part of the messenger RNA. So the result is that when these messenger RNA translate into proteins, in the proteins there are the amino acid sequences which are corresponding to these parts, they will be different from molecule to molecule. It means these class 1 chains which are made, these are alpha chains. These class 1 alpha chains which are being synthesized, in these specific points they will vary from molecule to molecule. Let me repeat it. It's a very important concept. As class 1 gene is constantly expressing, and before making the next set of messenger RNA, it will shuffle some of its genes. So when these messenger RNAs are being translated into class 1 alpha chains, within these alpha chains there is a sequence of peptide or sequence of amino acids which are varying from molecule to molecule. Because it is varying too much from molecule to molecule to molecule to molecule, we call that this within the gene and within this messenger RNA and within this peptide chain, these are hypervariable areas. What are these? Hypervariable area. You must be thinking, what's the use of making molecules with so different ways? I will tell you. You just imagine this molecule is like a human being. This molecule is like a human being, and the strange thing is that this is the machine which is making the human beings, and human beings are coming with same thing but different faces. Are you understanding? Here face is different, here face is different, here face is different. Is that right? You must be thinking, what is the purpose of this variability here? Right? Let me make one molecule big out of this. I've made one alpha chain of the class 1 large. Let's suppose this is this one, the first molecule. Now this is the porch. Okay, this alpha chain is said to be alpha 1 loop. Within the alpha chain, this is alpha 1 loop. This is called alpha 2 loop or domain, and this is called alpha 3 domain. So there is a pocket present between the alpha 1 and alpha 2. The pocket, this pocket or this groove, or this is the mouth of the molecule. This mouth of the molecule varies from molecule to molecule. Now why it is varying? There's a reason. Because we will see that later on in these pockets, these molecules will capture certain antigens. What is this? This is a peptide fragment. This is a fragment of peptide. These peptide fragment fit into these grooves. And if you are making these molecules with different grooves, right, it means different antigens will fit into these area. For example, the antigen which will fit here should be like this. Antigen which fit here, it is different. An antigen which should fit here is different. So what really happens? It's worth concentrating. What really happens that MHC class 1 genes are constantly producing class 1 molecules with hypervariable regions, and from molecule to molecule these pockets are varying in their amino acid sequence. Due to that reason, every molecule is capable of binding with different antigen as compared to the other one. Let me tell you, if one day within 24 hours in one of your cell, if 10,000 such molecules are made, you will be surprised to know that all these 10,000 molecules may be slightly different from each other in these regions of the molecule. So it means they are capable of binding with maybe 10,000 different antigenic peptide chains. Am I clear? Thank God these things happen automatically. We have to worry only for step 1. Is that right? There's so many things which are going on their own, and we sometimes forget to appreciate that. Right. Now there's one thing. This is a molecule having the mouth and fitting the antigen here or peptide chain here, right? Now we come to, this looks like, what is this? It is having 1 feet here, and what it looks? It is something like limping around. So nature has provided a sportive molecule here. To complete class 1 molecule, nature has provided an additional molecule here. This molecule is called beta 2 microglobin. This beta 2 microglobin bind with the alpha chain. Is that right? But very important point, beta 2 microglobin is expressed from chromosome number 15. The gene for beta 2 microglobin is coming from, yes, chromosome number 15. It means that beta 2 microglobin gene is not the part of MHC locus or MHC complex. Actually, whole this gene is, these 3 boxes together are called MHC complex. So this is, you can say, an additional support to that. Am I clear? Now, I hope you are very much clear about this mouth of this molecule. Where is the mouth present? Mouth is present in between the adjacent part of alpha 1 and alpha 2 domains of what? Alpha 1 and alpha 2 domains of alpha chain. Right? These are varying from molecule to molecule and determining the specificity for the antigen binding. Claro? Now we can talk of one more thing about this molecule. What is that? Okay. Actually, this gene is highly diverse. This gene, class 1 A gene, right? This gene has many alleles. This is polymorphic gene. It has many alleles. I think I need to explain it properly. Am I right? Let me tell you. Okay, Irene, you can come here. Alberto, you can also come here. And you want to come here? I want to give some example. Yes, please. His good name is Ramiro, right? Here is Ramiro, and Marla will also come here. I will explain few things, just a little bit. Leave the place so that, yes. Of course, there's no doubt these are 2 human beings, right? They are made of so many cells. And if I take some cells from Ramiro and I take some cells from Marla, is that right? And we mix them in a tray, with special test, I can tell with great confidence which cell came from Ramiro and which cell came from Marla. How I know it? Because as every human being has a fingerprint that is unique to him. By fingerprint studies, you can identify the human beings. In the same way, human cells have molecular fingerprints on them. Human cells have molecular fingerprinting on every cell. Because there are molecular fingerprints present on every cell, if you know what are these molecular fingerprints, what are special molecular signatures on every cell, if you recognize, you can tell which cell is coming from which person. For example, again I will repeat it. You take 10 cells from Ramiro, put into a culture dish. You take 10 cells from me, you take 10 cells from Marla and mix all those cells. A good scientist who can recognize the specific molecules on the surface of the cells, he can just pick one cell, he can do some studies. Oh, this cell is coming from Marla. He pick one more cell, look at it, make some studies, and then get, oh, it is from Ramiro. Or even he can find some cell, he say, oh, it is not from Marla, not from Ramiro, even not from Dr. Najib, someone has contaminated. This is a very unique quality. Every cell has its ID. Dr. Najib's every cell has ID of Dr. Najib. Ramiro's every cell has ID of identification of Ramiro, molecular IDs. And Marla's every cell has molecular IDs. Now what are these molecular IDs? These are class 1 and class 2 molecules. What are these molecular IDs? Class 1 and class 2 molecules. And now I will explain how these molecules work as molecular fingerprints. Right? What I'm going to say, it means Ramiro's class 1 molecules and class 2 molecules, the set of class 1 and class 2 molecules in Ramiro are different from my set of class 1 and class 2 molecules. And Marla's class 1 and class 2 molecules, which are expressed on her cells, are having some difference from my class 1 and class 2 molecules and from Ramiro's class 1 and class 2 molecules. Is that right? Now we'll see how class 1 molecules help or act as molecular signatures or molecular fingerprints. I will explain that. Right? Have a seat. Yes, you have a question.
Are MHC molecules inherited, from
We'll go to that. We'll go into detail of that, how they are inherited. First, we are just looking at what are these molecules. We'll talk about their inheritance pattern and different things for sure. Okay, let me tell you. Actually, if I remove one, this is the A, MHC class 1, A gene. We can call it A locus. What we call it? A locus. Locus is a point in the genetic material having a specific gene for specific function. Is that right? So this is one locus, this is another locus, this is another locus. Let's suppose I bring this locus out. And here I put a cell from Marla. Here I put a cell from Najib. And from here I put a cell from Ramiro. Najib, and I have to draw some diagram at the top, and Marla. I'll tell you how the specificity comes. If we only study A locus, of course Marla should have chromosome number 6. Right? I'm making the chromosome number 6, short arm, long arm I've cut. Now, all of us should have MHC genes here. Is that right? All of us should have A locus. MHC class 1 genes for A locus I'm only showing. Now a very important point. Actually, A gene, the gene which should be here, in the human, if you take A gene out of Marla, you bring A gene out of Najib, you bring A gene out of Ramiro, you will be surprised, maybe this A gene is slightly different from Najib's A gene. And it is slightly different from Ramiro's A gene. Maybe we can call the A gene which is coming out of Marla's disposition A1 gene, which is coming from Dr. Najeeb's A locus A2 gene. Here we call it A3 gene. Actually, if you study whole human population, if you study a big part of a human population, as far as MHC class 1 molecule gene at locus A is concerned, if you make a big study, you will come to know that there are many different type of A genes. Let's suppose A genes are from A1 up to suppose A50. They are still discovering, so no need to memorize their number. Every year they bring more discoveries, right? Let us suppose in the human beings, in a big, large part of a population, you bring the chromosome number 6 out from different persons and study the MHC class 1 locus, specifically at locus A, and you study only A genes, you will find A genes are slightly different from person to person, right? So these A genes are about 50. Is that right? So it means that every one of you at one chromosome number 6 should have A gene out of any number between 1 and 50. Maybe Irene has A39. Maybe Louis has A24. Maybe Israel has A14. Are you understanding? Am I clear? Now look. Marla has, let's suppose, inherited A1. Najeeb has inherited A2. And Ramiro has inherited A3. You have to remember that even though there are 50 varieties of A gene, but in one person at one chromosome number 6, you will have only one of those 50. For example, we put a cell of Louis. This is Mr. Louis cell, Louis Alberto, and we study the same study here in the A locus. When you bring it out, we come to know it is A23. But he will not have A1, 2, 3 or other A. So it means the varieties of A genes are about 50. And out of those 50 variant, you are going to have only one at one chromosome number 6. It means that one should be coming from maternal side and other should be coming from paternal side. So as far as class 1 molecules are concerned, right, as far as class 1 molecules are concerned, if you focus on the A locus, you will have one maternal A locus and one paternal A locus, one on maternal chromosome number 6, other on paternal chromosome number 6. And it is quite possible, or usually maternal may be different than the paternal. Am I right? What is the specific name used for these things? That you may have any one of these. What is the relationship of A1 with A2 and A3? They are called alleles of each other. What are they called? Alleles of each other. Alleles of each other. So it means for the A locus, there are many alleles, and everyone inherited one specific allele on maternal chromosome and other specific allele on paternal chromosome. Is that right? Is it clear? Now, how these are different from each other? How A1, A2 and A3 are different from each other? We have to see it. Actually, in this part of the molecule, this is another part of the molecule. This part of the gene varies in those 50 alleles. Again listen. This was a gene of class 1 molecule belonging to A group. I told you within A group what is happening, that, listen again carefully. Within the A group, if you study multiple different human beings, then A from one person may be different from other person at this point. For example, Dr. Najeeb has this like that. Maybe Marla has this like this. Maybe Ramiro has the same area like this. So this is a point which varies from person to person. Of course, this will be also translated into what? Of course, first this DNA portion will be transcribed into messenger RNA and eventually translated into protein in class 1 molecule. So in class 1 molecule, this is the point. This is the point in the class 1 molecule peptide, right, that you may inherit it, maybe this point, Marla has inherited A1. So her all, attention carefully, let's suppose this is Marla's nucleus. So she has A1 here on maternal side, and on paternal side she has suppose A7. Is that right? She is going to have A gene on maternal chromosome as well as on paternal chromosome. Here will be one allele, there will be another allele. Is that right? So suppose she has A1 allele here and A7 allele here. But when we talk about Najib, maybe he has A1, A2 allele on maternal side and he has A18 on paternal side. If we talk about Ramirio, he has A3 on maternal side, maybe A27 on paternal side. Is that right? Now how these A molecules are different from each other, I told you within a gene, now this is a different part of the gene. It is not the same part as hypervariable, which was determining the antigenic binding. This is a different part, and in this different part, what is happening that it varies from allele to allele. The right word should be this part of the gene varies allele to allele, and this keep on changing itself, but this does not change. Once you have inherited a specific allele, you are stuck with that allele, but this part keep on changing. So it means when this gene will express all the molecules, class 1A molecules which are made, if it is class 1A1, then all molecules will be A1. Now come here. About Marla case, I'm going to make class 1 molecules which are expressed by this cell. What we have said that she has, from mother side she has A, from mother side she has A1. On from paternal side, what did I say previously? You remember so well, A7. Right? Now, this gene will keep on making molecules which will express on the cell. This gene will also keep on making molecules, and those molecules will express on the surface of the cell. Is that right? Look at them. This is coming from maternal chromosome, and this is the molecule which is coming from paternal chromosome. And of course you know it. What is this? Beta 2 microglobin. Is that right? Now this is class 1A molecule synthesized from maternal allele. This is class 1A molecule synthesized from paternal allele. And of course they are different at these points. Maybe, now listen carefully, from this gene maybe 10,000 class 1 are made. From this gene also 10,000 class 1 are made. So how many class 1 are expressing here? 20,000. 10,000 coming from maternal gene and 10,000 coming from paternal. So on this one cell, suppose Marla has 10,000 class 1A molecule from maternal side expressed, 10,000 class 1 molecule expressed from paternal side. All the maternal molecule will be A1. And all the paternal molecule will be A7. Is that right? And if A1 has unique thing like this here and A7 has unique thing like that here, so it means half of the class 1 molecule will be of this category, half of the class 1 will be this category. Are you understanding? But within all the A1 molecule, if you study these pockets, they will be varying from molecule to molecule. And within all the class 1 A7 molecules, if you study this pocket, that will be varying from molecule to molecule. Are you understanding? So it means that Marla's secret number on her every cell is A1, A7. Now we talk about Ramiro. He has of course one maternal chromosome, another paternal chromosome. Here was A3, and what was there? Do you remember what did I say? A27. That's good. Now, When he will make class 1 on every cell or every nucleated cell, then platelets, he expresses thousands of the class 1. Half of them are coming from maternal locus, half of them are coming from paternal locus. Now, his class 1 molecules which are coming over here, now look, again, these are A3 molecules and these are A27. Of course, they should not be limping around, they should have their friend here, beta 2 microglobin. Now, here's half of those molecules which are A3 and half of those which are A27. This will be the point here, amino acid sequence which may be unique here. And amino acid sequence here may be unique here. Is that right? Now look at it. A3 is different from A1 and also different from A7. In the same way, A3 is different from A27 also. You are getting it? Now the special number on his cell can be as what was the Marla's number? A1, A7. So we can say Marla is A1, A7 as far as class 1A molecules are concerned. Ramiro will be A, yes, 3, A27 as far as class 1A molecules are concerned. Do you think now these molecules and those molecules are matching with each other? No. Is that right? And these are those differences that which our immune system can recognize. We'll talk about that later. Now, these are the molecular signature that you take any cell of Marla, you take any nucleated cells of Marla, right? It will have these molecules on its surface. And you take any nucleated cell of Ramiro and you will have these molecules there. And if you can recognize these molecules, you can tell which cell is belonging to Marla and which cell is belonging to Ramiro. Is that right? No problem up to this? Now, let's come back. We have said this gene has this portion which is varying from molecule to molecule. So all the maternal molecules or paternal A molecules will have these pockets different from each other, right? But all the maternal will have this area identical and all paternal will have in the same person identical. Am I clear? So it means, let's come back. This part of the gene is providing hypervariability to bind with different antigens. So this part of the gene is determining this pocket. And this part of the gene is determining allelic specificity. It determines the allelic specificity. So this will remain absolutely identical in all the class 1 molecule in one person coming from one chromosome number 6. Am I clear? Then we come to another thing. It has another domain here. It is derived, let's suppose, from this part of the molecule, gene. This is the third part of the gene. Now this third part of the gene, right, it is constant. It does not undergo any changes. It is constant in all what? All the molecules which belong to A category or C category or B category. Again listen. MHC class 1 A molecules or MHC class 1 C molecules or MHC class 1 B molecules. These areas are absolutely identical. Because these areas are identical in all the class 1 molecules in all the human race. All the human race. So we are calling them what? They are constant. They are constant. So all the class 1 molecules, either they are produced in Marla from maternal or from paternal, this region will be the same. Even class 1 molecules, whatever alleles are expressing there, but this area will be same in Ramiro's both type of molecules, but they will be also identical with Marla's class 1 molecule. They add these regions. So what does it mean? That when we are talking about any given class 1 molecule, we should talk about how many regions of that class 1 molecule? 3 regions. For example, this is a specific class 1 molecule. First region which I discussed was in class 1 molecule, first region is the groove made by the adjacent part of alpha 1 and alpha 2. This groove varies from molecule to molecule. Is that right? Then class 1 molecules have another, you can say, region. In that region, amino acid sequence varies from allele to allele. Is that right? Then another region in the class 1 molecule which is absolutely constant in all the class 1 in the given person, even that is absolutely identical, even class 1 molecules brought from different people. So if you take Marla's class 1 molecule or Najib's class 1 molecule or Ramiro's class 1 molecule, regardless of the class 1 molecules are coming from which person, and regardless of that class 1 molecules are A or B or C, whatever they are, they will always have these portions absolutely identical. You know why nature has made them identical? Because later on we'll study that when T cells, there are cells called T cytotoxic cells, they have a special molecule which is called CD8. This is called CD8 molecule. Actually, the CD8 molecule of T cytotoxic cells fits over here. So this domain of the class 1 molecule, right, is constant, and it is the recognition and interaction site between the class 1 molecule and CD8 molecules. Is that right? It means if T cytotoxic cell comes, it can fit with the any class 1 of A category or class 1 of B category or class 1 of the C category or whatever allele, CD8 molecule can fit over this constant regions. So it means we can say in the class 1 molecule, this portion is having a very special function of binding antigen, because different class 1 molecules bind with different peptide antigens or peptide fragments. So this area varies from molecule to molecule. Then we come to second area. In the class 1 molecule, in the second area, this area determines allelic specificity. In a given allele, on all the molecules, this area is identical. But in class 1 molecules coming from different alleles will have this area slightly different from each other. Then we come to this area. This area of the class 1 molecule is constant in all the molecules in 1% and also constant in all the molecules, class 1 molecules in different persons. And this area is specific for what thing? This area is specific for interaction with CD8 molecule. Am I clear? No problem up to this? Now, let's come out that this person, let's suppose here he is making A1. Here it is making C38. Here it is making B24. Because at every locus, as I told you in A locus, there may be any allele somewhere between A1 to A50, but there will be only one allele. In the same way in the C focus, there are also multiple alleles. In B focus, also there are multiple alleles. Again, let me tell you, if you think A is a box for a pigeon, how many types of pigeons we have? 50. But you have only one pigeon at one chromosome out of those 50. In the same way, if C is the monkey cage, how many types of monkey are there? Maybe 80. So any one of those may be here. How many types of B is for? Yes, pancake or ball, right? B gene is supposed like a ball. How many balls are there? Many types of ball. But in one given person, there will be only one ball here. Maybe ball number 28, maybe ball number 31, maybe ball number 19. You get it? So it means in any given human cell, how many types of class 1 can be there? Now I will make a bigger cell and express it. How many class 1 can be there on one given person's one nucleated cell? let's suppose I bring one cell out of Marla. Okay, we'll compare the cells of Ramiro and Marla. You never know, one day they get married and we'll see what happens with their kids. This is Ramiro and here is Marla. Right? Now we come to okay, I'll make it even more prominent. We'll totally disappear the long arm. This is long arm is down. This is only the short arm. How many chromosome number 6 should be present in every cell of Marla? 2. In most of somatic cells, of course. And in case of Ramiro, how many chromosomes number 6 will be there? Again 2. Right? Now listen. This is the box for class 1 molecule. And this is a box for class 2 molecules. And this is some class 3 molecules, we'll talk about them later. Right? And now box for class 1 molecule, how many regions it have? A, C and B. Is that right? And of course, this one, which one? Which was? Maternal. And here it will be paternal chromosome number 6. Paternal 6. Chromosome number 6 coming from paternal origin. Right? P0 for petite. And this is chromosome number 6 of Marla from maternal origin. On paternal chromosome number 6, she will also have how many loci? 3 loci. A, B and A, C and B. They are arranged like that. And of course, she will have genes for class 2 also. About class 2 we'll discuss later. Right now we just concentrate about the, and of course we come to Ramiro. Ramiro also has a mother, isn't it? So there will be a maternal chromosome on Ramiro, and here should be a paternal chromosome also. Okay, his father has a lot of hair, let's suppose. Right? He's a little bit likes French fashion. Right? Now, this is class 1 molecule 1A, C and B, and of course on the paternal A locus the A, C and B. Now listen carefully. We start with Marla. A was on maternal side A1 and paternal side we made A7, and we have discussed, suppose on Ramiro class 1 molecule on maternal side was A3 and on paternal side it was 27. In the same way, C will be also different. C may be here 19 because there are many C, but one of them will be fitting here. She has inherited from maternal side one of the total pool of C allele, and here is C21, and he has inherited C, let us suppose 32 and 43. And when we come to the B area, she has on maternal side B5, on paternal side B15, and Ramiro has B, which B here? Okay, 22 you like, you can choose anything, and B38. Now look, how many class 1 molecules Marla will have? First of all, we'll talk about, as far as these alleles are concerned. How many types of class 1 molecules Marla can have? Marla can have how many? Tell me. Yeah? 10,000. Oh my God. Of course, there will be not even more than 10,000. On a given cell, there are thousands and thousands of class 1 molecules. But all of them can be divided into two groups. Half of our all class 1 molecules will be of maternal side, and half of all our class 1 molecules will be on paternal side. And from maternal box, what are the three primary types of the molecule? 3. And for paternal box, how many types of the class 1 molecule will come? 3. So if we put the class 1 molecules, let's write down. This will be A1. Suppose these are the class 1 molecules. When this will express, A1 will come on surface, when this will C19, B, what is this? 5. These are all molecules from maternal haplotype. You know the concept of haplotype? Haplotype means the set of genes which are coming from one parent. So these three set of genes, A1, C19 and B7, these are Marla's maternal haplotype as far as class 1 are concerned. And these are Marla's paternal haplotype as far as her father is concerned. Is that right? From maternal haplotype of class 1 molecule, right, the set will be A1, C19, B7. So we'll say B5. So what we'll call it? What are these? These are MHC class 1 molecule from maternal haplotype or maternal chromosome. Clear? Then you will tell me about this. From paternal side, of course, paternal chromosome will also express. Listen, there is a word used. When these genes are expressing themselves, maternal genes are expressing themselves independent of the action of paternal genes. And paternal genes keep on expressing themselves independent of maternal alleles. Again, as far as class 1 genes are concerned, maternal alleles and paternal alleles keep on expressing themselves independent of the presence of each other. It means these genes are not, right, recessed by the others. They are dominant on their own right. And these are dominant on their own right. The genes which fully express, they are dominant. Is that right? Now, it means maternal alleles and paternal alleles, as far as class 1 genes are concerned, are independently expressing. Right? So we say these gene sets are co-dominant. What is the term which is used? Is they are co-dominant. So in Marla's molecular signature on her every cell, this is a maternal molecular signature. And what will be the paternal molecular signature here? Anyone who will tell me? A7, C21, very good, and B15, right? So what will be this? These three molecules will be class 1 MHC, class 1 molecule. These are of paternal haplotype. Oh my God, maternal haplotype or maternal alleles, and this is from paternal haplotype. Now listen carefully. On every cell of Marla, there are how many types of class 1 molecules expressed? 3 maternal types, 3 paternal types, in total 6. So every human being expresses 6 type of class 1 molecule on his every nucleated cells and platelets. And this is the special molecular signatures determined by class 1. Later on we will talk about that even class 2 also molecules add their own signatures on every cell of a specific person. Let's come to now Ramiro. We should not forget, he will get angry. So how many class 1 molecules on Ramiro? What types of class 1 molecules will be expressed on every cell of Ramiro? Yes. A3, C, 32, B, 22. Is that right? All these are MHC class 1 molecules expressed on in cells from which haplotype? Maternal haplotype. Is that right? And from paternal haplotypes you will have A, 27, C 43, and B 38. Now this is his set of paternal haplotype derived MHC class 1 molecules. Is that right? So Ramiro has on every nucleated cells how many types of class 1? 6. Now 6 are on the Marla cells, 6 are on the Ramiro cell, and are these molecules absolutely identical or different? It is just like as your ID card number, Marla ID card number is different, right? And Ramiro's ID card number is different. Is that right? Marla's telephone number is different. Ramiro's telephone number is different. Or Marla's every cell, set of class 1 are different combination. And types and combination of Ramiro's class 1 molecules are different. Is that right? Similarly, class 2 molecules also work. Is that right? They are also codominant. Okay, my friend want a break here.